Okay, so we're going to continue with the epithelium on this video. We have, after simple squamous that we ended with last time, we have simple cuboidal. So the cells are still only one cell layer thick. That's the tissue. I should say the tissue is only one cell layer thick. Here's our basement membrane. Basement membrane shown here. One cell layer thick. Here's our first contact. But now instead of being thin and flat, we're actually cubes, right? So a single layer of cube-like. That means there's more volume, more cytoplasm, more organelles than a squamous. So these cells would be able to do more functions, right? Simple cuboidal is found in a number of places. You guys are going to look at slides of the kidneys in lab to reinforce this idea of them. And of course, they are able to absorb, right, and secrete, and some protection as well, but really excel at absorption and secretion. So again, more space, more organelles means that they are able to make more chemicals, and they're able to process more chemicals. So you're going to see active transport in these cells, whereas again it was just diffusion or filtration for simple squamous. Facilitated diffusion, each one of these requires a carrier, and the cell has to make that. So I need more space. Simple columnar is also, just continues on, it gets a little bit taller. The nuclei, so here's our basement membrane along here. Our nuclei don't tend to be nice and round, now they tend to be oval. Here's the luminal area right here. These are actually, what you see are some microvilli on these. It just looks like a blur here, that's why they sometimes call them brush border. So single layer, again simple, tall, thin, rectangular cells. Sometimes they have cilia, sometimes microvilli. So in this case we're looking at microvilli. These are very commonly found in the digestive tract and lining that. So they're also going to be good at absorption and secretion, because with all this much more cytoplasm they have more organelles and they can process more chemicals. So again, you see these in the digestive, so they're good at making and secreting enzymes and mucus, and they're also all over in the small intestines, so this allows you to quickly absorb, get some food nutrient out here into the body just by crossing one cell layer. Okay, pseudostratified. So this is again falsely layered. So here as we look at this, when we consider a layer, what we're looking for, let's just go back here, is this lining up of nuclei all on the same basic plane. Let me go back one more. See that same thing here? All these are in a row basically. So we can tell that they're simple. There's only one row of these nuclei, right? But here in the pseudostratified, we've got nuclei down here, we have nuclei here in the middle, and we have nuclei up here. So it looks like we might have 3 layers of cells when we in fact only have one. And the reason for this is because the cells are not uniform in their height, right? And again, here's our apical outside. These ones also often have cilia, so much taller than the microvilli, and cilia are basically going to be moving things. So here again is our basement membrane. These are our epithelial cells. These are some mucus cells, so single glandular cells you find oftentimes associated with cilia, because mucus is very good at trapping things, but it's also a great food source for bacteria, so you have to move it along, right? So again, you find this pretty common in the respiratory tract, and they're very good at secretion, and they have some protective abilities, again, because you've got a protection from stuff out here to getting into the blood down here. Alright, stratified. So now we're adding layers, and we got some layers here, right? We can see all these nuclei in different rows, many layers here. And so stratified squamous, and while these, here's the outside world, if you will, and here's our body, deeper into our body. So while these layers down here tend to be kind of cuboidal, as we move toward the surface, the outermost layers are actually flat. So the tissue is named for the outer layers and what they look like. So we have stratified squamous. Again, this is a very protective tissue, right? To get to the blood down in here, you got to move all the way through all these cells. So this is an extremely protective. It's not going to function for absorption or secretion because, again, to absorb, I'd have to get it to the blood, right? And secretion, again, you'd have to be able to lead to the surface. So this is a protective tissue, all right? So where am I going to find it? Well, I show you here. It lines your oral cavity and pharynx, your esophagus, these digestive tract, and also the beginning of the respiratory there for the pharynx and oral cavity. All right. It is found in the anal canal. It's found in the vaginal canal, and of course, your skin is stratified squamous. So sometimes it's keratinized. That's where your skin is. So keratin is a protein that's a very hard protein, what your nails are made of. Right. So there's keratin in your skin, not in your oral cavity. All right. And the non-keratinized means is a moistureless, dry, and less hard tissue. So there can be some exchange. There's typically not exchange with the outside world across your skin. Okay. So stratified cuboidal, moving along that same thing. So now they're cube-shaped in more than one layer. Here we're looking at the salivary glands and the ducts that take. So this is a classic exocrine. I said it's not terribly common. Right, it's ducts of the sweat glands, some mammary glands, and salivary glands. Okay. Later on in AMP2, you look at the ovarian follicles, and they are stratified cuboidal, and the seminiferous tubules. So in the testes of the male. All right. They are more protective because again, here's the outside world. Here's blood down here. There can be some secretion, such as the ovarian follicles with estrogen and progesterone, and the seminiferous tubules, but they're not very common, and it's really more of a protective architecture. Stratified columnar, and you're not going to look at any examples of stratified cuboidal in lab or stratified columnar in lab. Okay, more than one layer, but only the surface cells are columnar. Deeper cells are cuboidal or irregular. So again, we're named for those surface cells that comes in contact. And here, the male urethra and a few ducts of glands. So ducts are the tubes that the secretion flows through to get to the surface of the body. Right? So they're really protective. Again, you're passing urine and semen through the urethra. So this is really, it would be found in here, and these are protecting. Right? So not a lot of layers like the stratified squamous, but they are layered. So we have to give them a different name. All right. Transitional epithelium, we may look at that in lab, but I probably won't test you on it on a lab practical. And of course, these tissues are going to be on your first lab practical. So transitional, it transitions in shape, as you see here and here, and in numbers of cell layers, right? So when it's not distended or stretched, like here, right, and here is a real one. And again, this shows you appropriately, it's really in on your bladder, parts of your urethra, and your ureters to bladder. There's about 5 or more layers of cuboidal or columnar, so nice rounded up. When you stretch, they become almost squamous. They just flatten out. So this allows your bladder to fill with urine, and you don't actually sense the volume change that's occurring in your bladder and in your ureters because this transitional epithelium changes to accommodate it. It just, again, if that doesn't make sense, just know for me that, again, transitional epithelium transitions in numbers of layers and in the shapes of the cells, so it allows expansion. And we'd find this in the urinary tract. Okay, so I always tell students, take a moment, think about what we've been talking about, and hopefully you are getting that, what the epithelial cells look like, helps us figure out what they do. So the foremost, again, function of epithelium is, since it's first contact, is the ability to interact safely with the world, right, on your terms, in other words, what your body needs. All right, so they control passage, they protect the structures beneath them, especially blood vessels, and they allow exchange. So again, you have to pass through these cells to get to the blood. All right, which means a single layer of cells are predominant in places where we need something to enter or leave, right? So again, secretion or absorption. Multiple layers where we really want to prevent any breaches, right? Any accidental entries, right? So very, very protective. and skin, oral and anal cavity. These are directly open to the outside world, which is a nasty place. Flat and thin, if I want diffusion to occur, because it's time dependent, right, as you guys did in lab. So here we'll see that in the alveoli, the renal corpuscles, and of course capillaries. Right? Cuboidal or columnar, if we need more organelles to create some, channels and receptors, and we need more mitochondria for more ATP. So if I'm moving things around, yeah, I need a bigger cell than these thin flat ones. All right? So before we go on to the next, to connective tissue, I want to say a little something about glands, because glands are secretory structures, right? They secrete things. So glands are primarily epithelium. I've already mentioned a little bit about salivary glands and sebaceous glands. So we're just going to come back and, kind of finish that up. Exocrine and endocrine. Now, endocrine glands have their own system. Exocrine do not. The word crine means to secrete. Exo is exiting to the outside. Endo is within. Okay? So exocrine, the other difference is that exocrine glands have to have ducts. They have to have these specialized tubular structures, and that helps deliver their secretion, whatever chemical that is, to the surface of the body. Endocrine, they do not have ducts. They're dumping their chemicals into the blood. So exocrine, they can be one cell, like the goblet cells you saw with the pseudostratified, or they can be many cells, like these cartoons show. They are organized by their shape and number of ducts. Yeah, I've seen a common theme. So if you only have one duct, you're simple. If you have more, and again the ducts are shown in the light color, the glandular tissues are darker. You have more than one duct, then you're a compound. And then you're just, what do you look like? All right? You're also classified by the way you secrete as a gland. Okay? So here's 3 strategies. Right? Merocrine, apocrine, or holocrine. And most glands are merocrine. Okay? So in this one, what we have is the cell is making, in each case the cell, again, it's a gland, it's making the chemical it's secreting. But if you're a merocrine secretor, you're basically packaging your chemicals into little vesicles, and the vesicles are moving to the plasma membrane, fusing, and exocytosis. Okay? If you're apocrine, you actually are losing a piece of your cell, right? So these cells are, their secretions are a lot more complicated for that reason, because it's part of it's cellular stuff. And if you're holocrine, the cell itself, the whole cell is dying and sacrificed. So whole cell, right, dying. And so again, the secretions are pretty complicated compared to merocrine. All right. So merocrine, exocytosis. Apocrine, lose part of the cells, and holocrine, the entire cell is lost. So most again are merocrine and are divided into serous or mucus cells. Then examples: holocrine are like the sebaceous glands. Apocrine are like the mammary glands. Okay, and merocrine would be like your sweat glands. All right, so we're not going to play cahoots here, because again, in the interest of continuing on with the video, we will pick up on the next video and start talking about connective tissue.